All right, it's that time. So I'm going to kick us off. Thank you all for joining us on this Monday afternoon for the first installment of our summer seminar series here for the Midwest Climate Adaptation Science Center. Today, we're going to have presentations from two of our Midwest CASC graduate fellows. As you can see on the screen and probably got a notification about, we are recording this webinar, so please keep yourselves muted while our presenters are presenting, and we will have time for questions at the very end of the seminar today. Afterwards, I will be sharing out the recording of this seminar via email. It'll also be available on the Midwest CASC website, so you're invited to see it there. We welcome you to pop questions in the chat throughout today's presentations, but again, try to stay on mute while our folks are presenting, and you'll have plenty of time to ask questions towards the end. Before we dive into things, I want to give a brief overview of who we are here at the Midwest Climate Adaptation Science Center. I'm Jess. I am the communications manager for the Midwest CASC, and we are a partnership between the U.S. Geological Survey and a consortium of 9 institutions throughout the region, which you can see the logos on the screen here, hosted by the University of Minnesota, which is where I am based. We're part of a national network of these regional CASCs, all of which work to produce science that can inform resource management efforts and help fish, wildlife, water, land, and people adapt to a changing climate. We accomplish this in several different ways. We conduct synthesis research on topics that are relevant to our region. We support a network of graduate students, including the ones that you'll hear from today. We partner with the College of Menominee Nation to run a tribal undergraduate research program in the summertime. We host events like these seminars, as well as our summer symposium, which I'll talk more about in just a second. And then, of course, the USGS funds climate adaptation research projects and partnerships through the CASC. Next slide, Alyssa. So as I just mentioned, we do have our summer symposium coming up this August. You're all welcome to join us there. Registration is now open. You can learn more at that link, and I will also drop it into the chat in just a moment. With that, next slide, I'm going to turn things over to our presenters. We have Narda Triviño Silva, who is a graduate student at Michigan State University, and Shotaro Shiratsuru, who's a doctoral student at the University of Wisconsin-Madison. And both these folks are Midwest CASC graduate fellows. We are really excited to learn about the work that they've been doing. So I'm going to hand things over to Narda to kick us off.
Thank you, Jess. Let me share my screen. Can you see it?
Yep,
now?
that's great.
Can you hear me? Okay, good. Hi, everyone. I'm gonna talk about my research in oak success under pine and using underlying mechanisms to develop novel silviculture practices. Oh, just give me a sec. Okay. Sorry. I'm gonna talk about oak success under pine and using underlying mechanisms to develop novel silviculture practices in the Lake States region. So first I want to talk about forest ecosystems of Michigan. They are unique and diverse forest ecosystems, especially because they have this transition from conifer-dominated boreal forest to hardwood-dominated temperate forest in the south. And I'm specifically interested in 2 groups. One is red pine and oaks, especially white oak and red oak. And Michigan holds the southernmost distribution of red pine and the northernmost distribution of oak, as we can see in these 2 maps of these 2 species distribution maps. So oak, there's like an oak species decline. oaks are like very important for ecosystem functions in the lake states because they provide mast source for they provide mast for wildlife, but they're also very valuable hardwood for the forest industry. And as I say, they are in decline, and this is because of poor regeneration and poor seedling establishments. And there are many reasons for this. Some of those are herbivory, like overbrowsing, competition with more shade-tolerant species, and fire suppression. However, despite this oak decline, there is successful oak regeneration under pine. So there's research and some observations that there is like higher seedling mortality of in under oak stands compared to pine stands, and this could indicate that oaks regenerate better under red pine. So there are many mechanisms and hypotheses that that could explain what's happening, and some of those are like abiotic conditions, like light, temperature, soil water content, or protection from late spring frost of the seedlings, maybe under pine stands. But also biotic conditions, like for example, soil pathogens are accumulating more under oak soils, or possible beneficial mycorrhizal associations under red pine. So red plantations in the Lake States region, they are a lot of them were the product of these reforestation efforts by the Civil Conservation Corps in the 19th century. Red pine is important for the wood industry in the Lake States region. And a lot of the plantations are mature, even age, and single species plantations. And these make them vulnerable to global warming conditions. And there's this increased signs of mortality and stress symptoms in the trees. And then there's also a lot of fungal pathogens outbreaks, like Heterobasidium root disease, diplodia, or Xerococcus blight, and bark beetles. So there is an interest in rotation of mature red pine plantations with oak plantations. So there's like this red pine decline associated with diseases, and this will help break the disease cycle. And also red pine may not grow well under future warming conditions compared to oak. So here on the right, we have our different climate scenarios for Michigan. Even in lower emission scenario or higher emission scenario, we'll have like warming. So the main objective of my research is investigate potential factors that are contributing to this greater oak seedling success under red pine than oak. So we are running different experiments. A lot of this is still going on. It's in progress. And I'm gonna share with you some results of each of these items. So the first one is we wanted to evaluate the red pine and oak soil feedbacks on oak seedlings for nutrients and microbiome in a greenhouse experiment. We also wanted to evaluate the abiotic factors like light, soil moisture, temperature, pH under red pine and oak stands. We also wanted to measure oak seedling growth and survival in red pine and oak stands, characterize our oak microbial associations, and we hope that with this information, we will be able to inform silvicultural guidelines to grow oak trees under red pine. So first, these are our field sites in the Lower Peninsula of Michigan. So we have 4 study sites with pear, red pine, and oak stands. This is Kellogg Forest, and they are in a latitudinal gradient. And then our northernmost site is Atlanta. And all these stands were 10 to 25% canopy coverage. So for our first item, our first experiment looking at the soil nutrient and microbial feedbacks, we collected soils from our pine and oak stands from our 4 sites. And we wanted to evaluate the red oak performance in growth in a greenhouse experiment. So we took our soils and we had 2 different experiments. We had the first one was our nutrient feedback experiments, where we sterilize our soil and we planted red oak in the 2 types of soils, pine soil and oak soil. And we have our microbial feedback experiment, where we use a live inoculum from our pine stand and our oak stands to grow red oak in the greenhouse. So when we look at our results of our nutrient feedback experiment and our microbial feedback experiments, the 2 different colors are our soil inoculums, different types of soils from oak or pine. We didn't see much differences in the effects of nutrients or microbial inoculum on our red oak growth. But if we look at other variables in our experiments, such as pH and other microbial roots like root pathogen abundances, we found that if we look at panel on the left, we have soil pH in our x-axis and total dry weight in our y-axis, and we see that the oak dry weight increased in pine soils, but only at low soil pH conditions. And then if we look at our root pathogen abundances, we found that our oak dry weight decreased at high microbial pathogen abundances, regardless of the soil inoculum. For our experiment looking at the field abiotic factors, we set up sensors in the field to monitor light availability, air temperature, soil moisture, and we also planted 1-year-old white and red oak seedlings in our adjacent red oak and pine plantations to monitor growth. So one of the most interesting results that we found, it was that under our pine stands, there was higher light availability during the growing season in 2 of our sites in Atlanta and Kellogg Forest. But we found that in Kalkaska and Roscommon, our oak sites had higher light availability than our pine sites. And we think that in these 2 sites, in Kalkaska and Roscommon, we had a spongy moth infestation and some trees were killed. So what we did was we offset our light intensity values at bud break, and then we found that pine still had higher light availability than oak stands during the growing season. And this is like an ongoing data collection, but these are some of the preliminary results for red oak growth. And we found that at Kalkaska and Kellogg Forest, our red oak grew better under pine than under oak. And in Atlanta and Roscommon, we didn't see much differences. And same thing happened for the white oaks. So in Kalkaska and Kellogg Forest, they grew more under pine than under oak, but in the 2 other sites, there was not much differences. And lastly, to look at the white and red oak ectomycorrhizal communities, we planted 5 pairs of white and red oak. These 2 oaks are co-occurring in our red pine and oak stands. And we harvested these seedlings after 1.5 years and look at and characterize the root fungal communities using Illumina sequencing. So we expected that different oak species may form distinct ectomycorrhizal communities, and they may have some sort of host specificity. And we found some interesting results when we look at our ectomycorrhizal communities. So we found that cover type, oak or pine type are shaping the root ectomycorrhizal community rather than the oak species. So if we look at this graph, we see that seedlings that were planted under oak, they have more similar ectomycorrhizal communities than the ones that, and we see the same for pine. They're more similar ectomycorrhizal communities, regardless of the oak species, if it's white or oak. So they had little host specificity between these two oak species for ectomycorrhizal fungi communities. And another interesting thing that we found, and we want to look more into this, it was that when we look at the ectomycorrhizal diversity, we found here on the y-axis, we have our Shannon index, a measure of diversity. And if we look at the upper panel, we have our 4 sides, and they're organized from south to north. And if we look at our red oak diversity, we see that when they're planted under oak, we see an increase in diversity with latitude. And we saw a pattern also for a white oak when they were planted under pine. So our ectomycorrhizal diversity decreases with latitude in our white oak. This is something that's still going on, and we want to look more into this. So some of the take-home messages from this presentation is that soil pH and root pathogen load determine oak seedling growth, at least in this system and in a greenhouse experiment. Also, that greater light intensity values under red pine plantations may be promoting better oak regeneration, and that diversity levels of oak ectomycorrhizal communities are shaped by latitude and cover type, and this could have implications for oak under red pine regeneration. And an example for this is pine soils. It's possible that pine soils won't support diverse mycorrhizal inoculum for white oak at higher latitudes. Yeah, this has to be further explored at the end of the research program. Yeah, and with that, I would take any questions, and thank you so much for your attention.
Thank you, Narda. So if anybody has a pressing question, we could do 1 to 2 right now, and then we're going to go ahead to the next presentation. But feel free to raise your hand or pop your question in the chat if you've got a quick one. I do see one in the chat, which is there much of a timber market for the red pine plantations in the Midwest?
Yes, it's still a very important species for the wood industry. Yeah, red pine.
Thanks.
Yep.
Okay, and then we'll take this one other one. Have you looked at the degree of mycorrhizal colonization among your sites as well? Do you think differences in the percent of colonization could impact your results?
Yeah, definitely. I think this is mostly from Illumina sequencing results. I didn't do any like microscopy work, but I think it would be interesting to look at the percentage of colonization, because it could give you different results. Yeah.
Okay. Thanks, everybody, and we'll have more time for questions at the end. show. I'm gonna put the spotlight on you.
Thanks. Yeah, let me share my screen first. Can you see my screen okay?
Yep, looks good.
Great, thanks. Okay, so hi everyone. My name is Shiratsuru, and I'm a wildlife ecology PhD student in Dr. Jonathan Polly's lab at University of Madison. So I've been a Midwest CASC Fellow for the current academic year. So I would like to thank Midwest CASC for their support and this great opportunity. So today I'm going to talk about my research, which aims to understand how the climate change alters activity patterns of winter adaptive prey species and thereby impact the predator-prey community dynamics. So in general, the prey species need to be active to eat and meet their energetic requirement, but at the same time, they also need to limit their activity to reduce the risk of predator encounter and predation mortality. So this food safety trade-off is the core idea in understanding the activity pattern of prey species, and my research is um, trying to understand the effect of warming winter conditions on this food safety trade-off in prey activity. So the warming winter has been impacting the winter adapted prey species by altering their interactions with predators. And I think the famous example is the snowshoe hare, which exhibits the seasonal coat color change induced by the photoperiod. So the black curve in this figure represents the change in percent white of snowshoe hares from fall to spring. So brown hairs turn fully white in like mid to late November, then start turning brown again in mid to late April. And the blue line in the figure represents the modeled snow duration from 70s to 90s. And then now the orange and red lines show the predicted future snow duration in mid-century for different emission scenarios. And the gray highlighted regions represent the coat color mismatch, where white hairs are predicted to be on the snowless brown background. So overall, the snow duration is predicted to be shorter in the future due to the warming winter and leading to the increased duration of the mismatch period. And the recent study from our research group showed that the snowshoe hares in Great Lakes region exhibit very limited plasticity in the timing of this seasonal coat color change. So these figures show the patterns of seasonal coat color change of snowshoe hare population in the Great Lakes region, which exhibits a very little regional variation on the mainland. And this camouflage mismatch makes it easier for predators to find snowshoe hares, leading to an increased predation mortality of snowshoe hares during these mismatch periods. And the previous study from our research group showed that the mismatched hares were more than like 3 times less likely to survive a week compared to the matched hares. And also the changing winter condition also negatively impacts survival of a winter adapted prey by facilitating the predator hunting success. And the research from Yukon Territory in Canada found that the snowshoe hare survival decreases with the decreasing snow depth. And this negative survival consequence of deteriorated snow conditions for snowshoe hares is caused by the increase in the hunting success of non-winter adapted generalist predators. So whereas the risk of predation by the snow-adapted predator Canada lynx is largely insensitive to snow conditions, the risk of predation by coyotes, which is a non-snow adapted generalist predator, increases with the decrease in the snow depth. So these effects of deteriorated snow condition induced by the warming winter has been causing the contraction of snowshoe hare's southern range boundary in the Great Lakes region. The previous study from our research group showed that the southern edge of snowshoe hare occupancy contracted from historical to contemporary time periods. And then their southern range boundary is predicted to shift northward even further by mid-century due to the lack of favorable snow condition. The regional extirpation of important food sources for diverse predators like snowshoe hares would significantly impact the entire ecological community. And so it is really important to understand the mechanism of predator-prey interactions under the changing winter conditions. So to understand how the predator-prey interactions would be altered by the warming winter, we need to acknowledge that the predators limit prey populations not only directly by predation, but also indirectly through the risk of predation or fear of predation. So like when prey species perceive the risk of predation, they will exhibit the anti-predator responses, like such as reduced activity or movement and increased vigilance and so on. And then in these anti-predator responses, the prey animals are trading off energy intake for safety, which can lead to energetic deficits. The prey animals are required to balance this food safety trade-off to optimize their fitness. And this food safety trade-off becomes more pronounced in the winter because of the low food availability, harsh weather conditions, and high risk of predation. So the warming winter conditions are expected to significantly impact this food safety trade-off and result in prey behaviors because the warming winter temperature can affect their energetic costs, and whereas the changing snow conditions can impact the risk of predation. And so one of the main questions my research tackles is how prey species optimize the balance of this food safety trade-off while like dealing with so many different types of risk at the same time in the natural environment. And the different types of risk for snowshoe hares include the immediate predator encounters, and then also environmental risk cues such as moonlight, strong wind, and snow condition, and then also the camouflage mismatch induced by the shortening snow duration. And additionally, as the winter progresses, the predation risk they experience accumulates over time, while their nutritional conditions deteriorate due to the food shortage. And both of these, like overwinter changes, further complicate the food safety trade-off. So to answer this type of question, we need to focus on the single prey species in a single ecosystem and to study every single detail of their activity with changing risk and environmental conditions. So the core data of my research is detailed behavior data obtained by the wildlife tracking collar deployed on wild snowshoe hares in the Shawinigan National Forest in Wisconsin, where the diverse predator species are present. So the collar is equipped with two different data loggers. One is a GPS unit, which collects the animal location data like every few hours. And the other is the accelerometer, which record body acceleration of animals every second. And these detailed behavior data tells us what animals are doing, where and when they're doing it, and as well as like how animals are altering their behaviors in response to variable risk and environmental conditions. But for today, I would like to focus on the activity data collected by the accelerometer. So the accelerometer records the body acceleration along the three different axes every second, and the different behavioral categories such as like running, walking, feeding, and resting would exhibit the different patterns of acceleration. And by combining these acceleration data with the behavioral observation of colored animals, we can classify the acceleration data into those distinct behavioral categories. And like this will allow us to tell like how much time per day animals spend feeding or resting, and how animals are like altering these patterns in response to the changing external conditions. And in addition to these behavior data from accelerometers, I conducted a field experiment over the winter to assess how additional risk factors affect snowshoe hare behaviors. So I set up like 4 experimental patches with automatic rabbit feeders in the study area. I paired up those rabbit feeders with trail cameras, which recorded the short videos of snowshoe hares that were attracted to those feeders. And based on these videos, I could quantify how much time snowshoe hares were spending being vigilant while they're feeding. And additionally, these videos showed whether snowshoe hares were exhibiting the camouflage mismatch or not. And also to manipulate like the long-term scale predation risk, I conducted the predator attraction over the winter in 2 of those 4 experimental patches by deploying the frozen beaver meat with the set lure. So the behavior data from the accelerometer collars show that the snowshoe hares reduced their foraging activity when the moonlight was bright because of high predation risk. And this response was more pronounced in the late winter. And similarly, when wind speed was high, the snowshoe hares reduced their foraging activity due to the high predation risk. However, this response attenuated from early to late winter, likely because of the nutritional condition of snowshoe hares deteriorated as winter progressed, and that led to the high foraging needs in late winter. And also the videos recorded by the trail cameras in the experimental patches showed that snowshoe hares increased the level of vigilance while feeding under the bright moonlight, and then because the highest predation risk. And this response attenuated from early to late winter. And hares also increased their vigilance when the wind speed was high, but this response did not change from early to late winter. And overall, the snowshoe hares reduced the level of vigilance from early to late winter, and this indicates that the snowshoe hares were nutritionally stressed in late winter, and they prioritized food intake over risk avoidance. But however, the snowshoe hares in the predator attraction patches maintain the high level of vigilance compared to the snowshoe hares in the control patches over the winter. And then this implies that the snowshoe hares still prioritized risk avoidance in late winter when they were exposed to the long-term high predation risk. And notably, the snowshoe hares exhibiting camouflage mismatch were more vigilant than non-mismatched hares. And this indicates that the camouflage mismatch can induce the reduction in food intake of snowshoe hares and potentially leading to the negative fitness consequences. So to summarize, hares are fine-tuning their behaviors in response to the diverse types of risk, and including the dynamic environmental conditions. And hares increasingly prioritized food acquisition over risk avoidance from early to late winter due to the increasing foraging needs. But this pattern was less pronounced when they were exposed to the high long-term risk. And again, notably, the camouflage mismatch resulted in the less feeding efficiency, implying that the camouflage mismatch caused by the warming winter can lead to indirect negative fitness consequences in addition to the increased predation mortality. So these results made me start thinking like how differently like winter conditions impact the food safety trade-off in different prey species. Like even in a single ecosystem, different prey species possess like different traits which can significantly impact the balance of the food safety trade-off. Like for example, the snowshoe hares and the North American porcupines all occur in the same ecosystem across the northern North America, and they share the same predator species. But like the porcupines are morphologically protected by the thousands of quails, which reduces their vulnerability to predators. And this would result in the lower level of risk perception by porcupines and compared to snowshoe hares. In addition, while porcupines possess the fat reserve from summer to fall, which they can rely on during the winter time, snowshoe hares do not have energy reserve. Therefore, hares are required to constantly forage, whereas porcupines can minimize their activity in the wintertime. So this will further relax the food safety trade-off for porcupines compared to snowshoe hares. So to answer this type of question, we need to compare the activity patterns of different prey species in the same ecosystem. So I compared the accelerometer-based winter activity data from snowshoe hares and North American porcupines in Wisconsin. So this figure shows that the relationship of activity time between two different snowshoe hares on the same day. So you can see the strong positive relationship, which shows that the different snowshoe hares showed very similar activity pattern under the same environmental conditions. In contrast, the different porcupines behaved very differently under the same environmental conditions, showing like a large individual variation. So these findings show that the porcupines are much more flexible in their activity compared to snowshoe hares. So to see if the maintenance of energy balance is the main driver of their winter activity, I used an energetic-based model to predict the daily activity time of both species. The model I used was based on the assumption that they optimized daily activity time so that they could maintain the energy balance. And the model considered the energy intake and energy expenditure of activity, as well as the thermoregulatory cost at low temperatures. So you can see that both hares and porcupines were much less active compared to model predictions when temperature was low. So this shows that both hares and porcupines were in the energy conservation mode and minimizing their activity on those cold days. And then even when temperature was mild, both species exhibited like large deviations of activity from energy balance. And notably, the environmental condition did not impact porcupine activity at all. But in contrast, activity of snowshoe hares were largely explained by the environmental factors such as wind speed, snow depth, and moonlight. Moreover, porcupines exhibited a larger individual variation and flexibility in their activity compared to snowshoe hares. So in snowshoe hares, like 20 km/h of increase in daily wind speed can result in 12.3% of reduction in their daily activity. This can lead to 7.4% of reduction in their daily net energy gain. And then also the 20 centimeter increase in daily snow depth have the same effect on their activity time and energy gain. So in summary, morphological protection and fat reserve relaxed food safety trade-off in porcupines and leading to the more behavioral flexibility in porcupines. In contrast, hair activity was largely explained by environmental risk factors, and their behavioral flexibility was highly limited. And reduction of hair activity in response to those environmental factors was predicted to result in non-negligible energy deficit. And then these findings suggest that the prey traits can mediate the food safety trade-off in the winter prey activity. So I think my research provides some broad takeaways for assessing the impact of the climate change on ecological communities and for the management plans. The first, my research showed that energy safety trade-off is a potential framework for evaluating the climate resilience of winter-adapted species. I found that the behavior of snowshoe hare is highly constrained by environmental conditions, whereas the behavior of porcupine is largely insensitive to winter conditions. Therefore, the hares are expected to be much more sensitive and vulnerable to changing winter conditions compared to porcupines. And second, my finding that the camouflage mismatch reduced feeding efficiency of snowshoe hares highlights that the warming winter conditions can impose indirect fitness costs on winter-adapted prey species. And I think this is important perspective for better predicting the impact of climate change on the populations of the winter-adapted species, because previous studies and management plans mostly focused on the direct effect of climate change, which is an increased mortality of those winter-adapted species. And finally, I also recommend that the management plans for winter-adapted prey species should consider the both aspects of energy safety trade-off. Like for the safety aspect, managers can either promote habitats that provide a protected refugia or actively control the number of predators. And for the food aspect, the managers can provide the species with better foraging habitat in winter to promote the fitness of the species. Again, I would like to thank the Midwest CASC for their support. And also I would like to thank all the logistical support I've received from our collaborators. Thanks for listening, and I'm happy to take any questions.
Thank you, Sho, and thank you to Narda as well. You guys both give great presentations. I'm gonna pop you both up on our screen and invite everybody to ask questions. You're welcome to raise your hand in the raise hand function so I can see you, or pop your question in the chat and I'll read it out loud to our presenters. I know we already have a couple in the chat, so I'm gonna try and catch up on those. One that just came in for Sho, what would be the optimal protective refugia and winter foraging plant communities to encourage through management?
So I would say, at least based on the previous study, so the young aspen stands would have a really have a beneficial effect for snowshoe hares to reduce their increased mortality caused by the camouflage mismatch. And I believe that snowshoe hares are also browsing those like young aspen stands. So I would say in general, like young aspen stands, like thick young aspen stands would have a huge benefit for snowshoe hares for protecting them.
Thanks. There's another one in here, which is in terms of impacts to behavior, is wind speed necessarily related to predation risk, or could it be related to thermal regulation?
Yeah, I think that's a really good question. When I analyzed my activity data, I included the temperature in the model as well. So which means the temperature effect was accounted for. So even when the temperature effect was accounted for, the wind had a significant impact. So I think there's still a possibility that there might be some like interaction effect between wind speed and the really cold temperature, but the wind speed per se has probably the independent effect, and then that is associated with the increased predation risk because of the polluted auditory cues and then also some chemical cues as well.
Thanks. One for Narda. Are there any plans to burn the sites that you were evaluating in your study, and do you think this would impact your results?
There's no plans for introducing fire in my experiment. And oaks are like fire adapted species, and I think it could have implications if you included fire, but mostly for natural regeneration of oak. So when you include fire, this will help like reduce competition with other types of vegetation, so oaks can germinate. But in our case, we germinated out seedlings in the greenhouse and we brought them to the field once they were germinated. So fire is not gonna be included in my research, but these are like DNR sites. So I don't know about the DNR plans for introducing fire in this specific stance. Yeah.
And then one more short one for you, Sho. Did you consider bobcat as another predator pressure in your research?
Yeah, that's also like a really great question. So when I was like starting my research field work, I was expecting to see more bobcat effect on snowshoe hares. But like throughout my the study period, my trail cameras only detected like single bobcat detection over the entire winter and the whole study site. So looks like the bobcat was not very active in my study site. Um, so, but that's a really good question.
Okay, I've run out of questions in the chat, so I invite folks to raise their hand. Oh, here's one. Narda, do you have any thoughts about how your research findings apply to practices like assisted migration? It sounds like soil or mycorrhizal communities could greatly impact success. Any thoughts on that?
Yeah, there's definitely a field of research looking at the how maybe different root traits or when you select for root traits and if you move species if they will couple well with the microbial communities in the soil. But yeah, there could be, I think that there could be some like uncoupling of like root traits or soil microbial communities or any trait that you select to um, um, and if you move species up, then maybe there can be some decoupling in this because they are mycorrhizal associations are like obligate symbionts, so they need a live host. So I think there could be something important there to look at and to consider when migrating or moving species from place to place. Yeah.
Thanks for sharing that comment in the chat, Tyler. That's cool.
Yeah. Thank you. Thank you for sharing that.
Okay, any additional questions? Again, if you want to raise your hand and ask your question directly or make a comment, you are welcome to. Yeah, I'm gonna wait another minute or so before I wrap us up, just because I know sometimes it takes a moment.
I'm going to take myself off mute. Hi, it's Alyssa. I was just wondering if you could both comment on either the next steps for the research or how you might be engaging or how you might be engaging practitioners in those pieces. I know that you've got several partners on the group, so maybe the first question is just kind of next steps. And maybe that's what you're doing, or maybe if you're not, what you're teeing up for others to do.
I can go first. So yeah, I'm my next steps for my research. We have several years of data collection. So this is like the first year data collection. I'll keep collecting data and kind of harvest my seedlings at the end of this year, and then kind of put all the pieces together with the final oak growth. Yeah, that's kind of like the next steps for my research. What was the other question you had?
If there were next steps too for engaging other partners in the research and those next steps.
Yes, so part of my research involves like inform silvicultural practices. So we would like to kind of compile this information and to for different types of stakeholders that are interested in managing red pine for oak regeneration. So we want to, besides doing like our publications and all of that, we want to have like an accessible information for different types of stakeholders in the region.
Okay, thanks.
Mhm.
Sho, do you have something to add for your
Yeah,
project?
sure. Yeah, so today I only like focused on two of my chapters, and the one was focusing on snowshoe hare in Wisconsin, and the other is comparing the snowshoe hares porcupines in the Wisconsin. But I'm currently trying to finish up my third chapter in which I compare those like activity patterns of snowshoe hares in the winter across three different sites throughout the northern North America. Wisconsin and Isle Royale National Park, and also Yukon Territory in Canada. So that research aims to understand how the differences in the predator species and also how the differences in the winter condition can impact their behaviors and the consequences. So I think that would provide some different perspective. Um,
Thank you.
Yeah, thank you to you both. Anyone have any final questions? Well, you guys both gave great presentations. So cool to learn about your research. Excited to see what comes next. Thank you to our presenters and to everybody for spending this time with us. As I said, this is the first installment of our summer seminar series. I'm going to put a link in the chat to that links to registration for the other installments. We'd love to see you in June and July. And then finally, I'm going to pop up a quick closing poll. Please take that before you hop off to let us know how we did and how we can improve. And finally, one last big thank you to you all. Thank you for sharing your work with us, and enjoy the rest of your Mondays.